This lecture is an important part of the Victorian Assisted Reproductive Treatment Authority's discussion program. One of the key roles of the authority is to provide education about issues relating to fertility and the prevention of infertility. And we do this in a variety of ways, including through the provision of information, community consultation, and encouraging discussion of the issues and the developments which arise in this area to ensure that people are well equipped to make informed decisions about planning and creating a family. Through the lecture series, we aim to introduce speakers who will inform and provide thought leadership for these discussions, providing an opportunity to reflect on the issues that arise from the use of assisted reproductive treatment in our society, and to tackle some of the questions that arise as technology and society constantly changes and innovates. The authority also conducts public information campaigns, and in this regard I can't resist the opportunity to advise that along with the Robinson Institute, Andrology Australia and the Jean Hales Foundation for Women's Health, we have formed the Fertility Coalition, my mother would be horrified, to develop a public information campaign called Your Fertility, to educate about the lifestyle factors that affect men's and women's fertility. Your Fertility has been funded by the Commonwealth Department of Health and Ageing through the Family Planning Grants Program. And we're currently in the planning stages of this initiative, and more information will be available over the next couple of months. In case you hadn't noticed, tonight's lecture is being filmed, and this will appear as a podcast on the Your Family website in due course. So start thinking of some insightful probing questions now, and you'll be there for posterity. Our presenter this evening, Professor Robert Norman, is also involved in the Your Fertility project, and the topic of his lecture is very relevant to the issues that will be highlighted through the initiative. So I'm accordingly delighted to welcome Professor Norman to deliver the Louis Waller Lecture this year. Professor Norman holds a personal chair as the Professor for Reproductive and Periconceptual Medicine at the University of Adelaide, and is a subspecialist in reproductive medicine and in endocrine biochemistry. Professor Norman is a director of the Robinson Institute at the University of Adelaide, a collection of over 350 researchers in reproductive health and regenerative medicine. He serves on the National Health and Medical Research Council's Research and Embryo Licensing Committees. He is leader of its program grant on periconceptual origins of health and disease, has published a book and around 300 peer-reviewed publications, and serves on the editorial board of a number of major journals. His major research contributions have been in IVF and reproductive endocrinology, particularly in polycystic ovarian syndrome, the effects of lifestyle on reproductive outcomes, and periconceptual medicine. He is an active reproductive medicine specialist. Ladies and gentlemen, would you please welcome Professor Rob Norman.
Thank you very much. I need to get my audio-visual sorted out first. Well, thank you very much to many of my friends and colleagues, and to the Melbourne community interested in reproductive medicine. It's a very great honour for me to present this, and I've been to one previous Waller lecture, and that was presented by Alan Trounson, and for me to be in the company of many predecessors who've given lectures, it's a wonderful thing. I'm going to take a view tonight that it's really important to know what reproduction can do for our future health. So I'm going to talk about why reproduction is important, if you didn't know that already. I'm going to tell you about 3 revolutions that are occurring in our society today that are going to radically transform the way that we look at reproduction and development. I want to talk about how our lifestyle, the decisions we make, or the government makes, or occurs in nature, impacts on our health and families' health. I want to talk about what we can do about this and what we should be doing now. And finally, I want to emphasize where we should be investing, where our time, our money, our interest should be going. So over the next 35 minutes, I hope I'll give you enough of a sample for you to be interested in this area. We clearly can't go into vast detail. And I want to keep it fairly general because I believe that it's an audience that would want that. But there will be some areas where I do get into more detail. So let's talk about reproduction. Those of you who are gardeners will know that reproduction is remarkably successful in nature. You're constantly pulling weeds out or killing mosquitoes or flies. So nature has evolved a way whereby reproduction, regardless of your intelligence or social status, is incredibly successful. And I can't think of a better poem or song other than the one that Paul McCartney put out. Though it takes a lot of power to make a big tree grow, it doesn't need a pot of knowledge, for a seed knows what a seed must know. So inbuilt into our whole status is a system that works most of the time without us understanding how it works or having to do very much thinking about it. So reproduction is generally remarkably successful. And reproduction is absolutely critical because in each person in this room, we have two developmental histories. One history is that goes back millions of years, and that is of our genes and how our genes have been transformed through natural selection and other processes, so that each one of us is a product of millennia of development. But each one of us is also a product of another form of development, and that is what happens when we're conceived. So from the moment that a sperm and an egg get together, the environment and our genetic component determines what we are as well. So together with evolution, we have the shorter term, which is what happens to us in our mother's womb and after we're born, and how that has a major impact on the sort of people that we are and our health. So let's remember that reproduction is fundamental to the paradigm of evolution. You can have the most wonderful genes, but if you can't reproduce, they're gone forever. And you can have pretty horrible genes, but if you can reproduce very successfully, you might be able to get those genes to keep going. Our developmental history is one that we don't often talk about in terms of evolution, but from the time that we have an embryo growing through to the time that we have a baby that's born, enormous influence has been brought to bear from the environment and from the impact of other things upon our genetic history. So reproduction is absolutely essential for our development as people. Clearly, genetics is important. You've got to have good genetics to produce a good child. There's the concept of fetal origins, that what happens to you in your mother's womb and the influences that she's exposed to also have a big impact on you as a person. Early nutrition, whether it's through the placenta or through mother's milk or postnatal nutrition, has fundamental impacts on our health and behavior. We have a life course of epidemiology, which is a discipline in itself, the sort of background that you grow up in, your social status, etc. The way the brain develops is critical because the brain is exposed at many times during development to episodes that may damage or promote it. And the way that a child or an embryo grows psychologically is also essential. So by the time we reach the age of 3 to 5, almost all of these things have had a major impact on the potential for health, for well-being and behavior for the future. And it's been proposed that this is the best investment that we can make in anybody's health. And economists have shown very convincingly that if you want the best return on human capital, it's much better to invest in a child under the age of 3 than it is in someone after school. You also get very good investment in preschool programs and in schooling. And as you get older and older, you have to put more and more money in to get the effect. So from an economic point of view, it makes a lot of sense to concentrate on this area of life because you can spend a small amount of money to get an early intervention and go on to get a person that has its full potential. Whereas if you invest later in life, as a teenager or as an adult, you've got to put a lot more money in to deal with things like poor parenting, drug and alcohol abuse, violence, teenage pregnancies, many of which may have their origins very, very early in life. Let me bring this back even earlier. I believe that if you invest even earlier, before pregnancy, you will get great results. And I'm not sure which one is there's laser printer. So here we have pre-pregnancy and pregnancy. And I'd like to take that same graph that I showed you and suggest that if you invest in sperm and embryos to get the best quality before they even produce an embryo, that's an even better investment. Pre-implantation, making sure that this embryo is conceived in an optimal environment. And then what goes on in the first trimester of pregnancy. So let me illustrate it this way. Let's say that you've got good genes and a good environment, that over time you're going to follow a trajectory like this, that you fulfill your full functional capacity, and only later in life does your capacity fall off and you develop disability or illness. If you start badly and you're on the wrong trajectory, you are going to apparently be a healthy baby, but you may enter disease and disability much earlier in life. It's getting this trajectory right that can only occur at the beginning of life and not later in life. So this is the concept of the periconception period. And we think that the periconception period probably goes back at least 12 weeks, because that's when sperm are formed and when eggs start to get selected, right the way through to when organs are developed, probably about 8 weeks. So during this time, the embryo is maximally exposed, and the sperm and the eggs, to environmental stimuli that are going to benefit or harm it. So what are the issues that start at conception? Well, clearly, if you can't get pregnant, if nothing's happening, that is an important thing to look for. Over 20 to 30% of miscarriage, of pregnancies overtly miscarry. So the woman will know that she has miscarried. There are a number of adverse pregnancy events that can go back to the very early implantation of the embryo. And this includes blood pressure, diabetes, congenital abnormalities. Preterm labor is closely tied with poor implantation. And then we have developmental abnormalities of intelligence and behavior. And you've probably heard this week about older men are much more likely to produce children with schizophrenia and autism. And that's been known for a long time. But recently genetic changes have been shown in older mice that may predispose to this. And then we have the whole concept that health and disease has its origins early in life. So these are important issues. Now, when we look clinically at spontaneous pregnancies, it's really a bit like an iceberg. We have live birth rate and we have miscarriage rate, and these are all recognized. But under the water, and often not recognized, are the fact that many embryos are conceived, but only implant for a couple of days. And we think also that many embryos are conceived that never implant. And I won't go through the evidence for this, but in humans, it's quite strong. When we look at IVF and assisted reproductive technology, the iceberg's perhaps a little lower. And we do get live birth and we do get miscarriages, but it's likely that this is much more common in IVF. We put beautiful embryos back and we just don't get a pregnancy. And it's possible that many of these events are occurring. If we could understand what's happening very early in life, we would perhaps be able to push the IVF iceberg up and the spontaneous conception iceberg as well. So let me go on to the convergence of new technologies. So we're living in exciting times. We live in a reproductive revolution that really originated with the contraceptive pill, but has washed through now into IVF and into stem cells and many other areas. We live in a genetic revolution that overtly started with Watson and Crick and determination of the structure of DNA. But now for $1,000 you can get your whole genome sequenced. And we live in an information revolution where we can take large quantities of data and manage it in unbelievable ways and in very practical ways, like doing a Google search on why you're not getting pregnant or what your IVF doctor has forgotten to tell you about. So all of these are converging together and are having a major impact on the way that we practice reproduction, and we're looking at the start of life. So let's start with reproduction. Well, many steroids were synthesized in the 1930s and 40s and 50s, but it really took the contraceptive pill to change the way that the general public looked at reproduction. The advances in reproductive biology led to IVF and all the changes that occurred there. So initially, we wanted to completely understand and control human reproduction. But now we want to move beyond that. We want to benefit human medicine generally, not only in health and disease in individuals, but also now through stem cells and regenerative technology, whereby we are going to be able to do cell therapy for people rather than drug therapy. So the reproductive revolution has washed far beyond people just getting pregnant. The technologies exploded as well. And if we start off with the contraceptive pill, you'll see that there are a whole series of technologies. The dates may not be entirely correct, but certainly on the first part of the spiral, Melbourne and Victoria were it. Everything was happening here. And amazing discoveries that go down in history as Victoria being the first. So many of these things occurred in the 1980s. We were the first to get a pregnancy in Adelaide from pre-implantation genetic diagnosis and also from sperm injection. And these were only the third groups in the world to get pregnancies. These were the first groups in the world. And we've got great blastocyst growth now. And in Melbourne, I think you're going to lead the world in doing a new form of pre-implantation genetic diagnosis. So the technology's exploded, the law has not followed, and people are very confused about the ethics. So what's happened across Australia? Well, in blue are the total number of births every year, and that's shown in this axis here. In green are the percentage of IVF babies that are being born, and the figures that are going to be released in the next couple of weeks show that we're up now at 4% of all babies are born from IVF. And if we take Israel and Denmark and others as an example, this is only going to go up and up. So IVF is being used more and more. IVF pregnancy rates continue to go up. And I've been involved with clinics for the past 25 years. And this shows you the pregnancy rates of the clinics that I've been involved in for women 38 and under. And it's expressed as a fetal heart per egg recovery. And it's important to know what people are using. And you'll see that for many years, we didn't get very far. We got pregnancies. But we were introducing lots and lots of new technologies in there. And then from the year 2000, you'll see that the pregnancy rates started to climb again. Was this because we were putting lots of embryos back? Well, this is the implantation rate, which is the chance of one embryo forming a baby. And you'll see that there was a real increase in efficiency that occurred over the past 10 to 15 years. And as a result of that, in red, are our multiple pregnancy rates, which used to be triplets and twins. And John McBain was one of the world leaders in telling us this was totally unacceptable. And you'll see here that around about the year 2000, our multiple pregnancy rate started dropping, and last year was 4%. And the number of embryos that we put back at one stage was 4 or 5, and it's now down at 1.1, whatever that means. So our IVF pregnancy rates are getting better and better. And our fertility options have changed. I guess the traditional approach was to make a diagnosis, advise patients, come back in a couple of years, do simple things, possibly use advanced technology such as intrauterine insemination a bit later. The current practice, which in my view has gone too far, is forget about the diagnosis, come and have an IVF cycle, you'll get pregnant quickly, and by the way, we don't want to take the chance that you won't fertilize your egg, so we'll inject the sperm. And I think that that is the opposite extreme to where we should be. But certainly, having gone from the stepped-up pathway, which many of the older gynecologists and practitioners have practiced, and I think still do practice, and I'm saying that in a positive way, they're now, particularly among the younger practitioners, is to go from see the patient, do IVF. So we're getting big changes in the way that fertility options are being managed. And we have reproductive menus. I have patients who come to me who know absolutely what they want. They know where they're going to go in India or Russia or South Africa to do things. And this is not just in IVF. People know what contraception they want. They have a right to termination of pregnancy. They would like to have babies after the age of reproductive life. They want to store sperm and eggs for the future because someone's told them that their ovarian reserve is terrible. They want to use donor sperm and eggs and embryos, which can be obtained in various places. We can't in Australia do sex selection of embryos, but that is something that's practiced elsewhere. Genetic diagnosis of embryos, personalized stem cells, surrogacy, and sexual rejuvenation using lifestyle drugs. So these are on the reproductive menu. And as a clinician, I get patients coming every day telling me that I'm out of date, I'm the waiter, they're prepared to pay for the dish, but I've got to order it from the menu. So patients are taking things very much into their own hands now. That's the reproductive revolution. What about the genomic revolution? Well, for the past 50, 60 years, we've understood the structure of DNA. In the past 10 years, we understand the structure of the human genome. And we now have chips in which you can commercially send your blood off or your cheek cells and get your whole DNA profile done. And we have a huge number of technologies that are available to us. So when it comes to reproduction and the earliest stages of life, there's some things that are pretty classical. So if you come from the Mediterranean and you've got anemia, you're likely to be checked for thalassemia before you become pregnant. And then hopefully you're going to find a partner that doesn't have the same gene as you. If you've got cystic fibrosis, if you've had a child with cystic fibrosis, then you would want to check that before you had another pregnancy. And if you're an Ashkenazi Jew, you'd want to know you've got Tay-Sachs disease gene or not, so that you can avoid marrying someone else with those genes. Increasingly, though, we're looking at variations in the genetic code, not abnormalities, but variations. And it appears as if some variations are predictive of disease and treatment. So, for instance, the FSH receptor, which is the receptor for the common hormone that we use for IVF, has variations. And if you have a particular type, you're going to overstimulate seriously. And it will be that for things like cerebral palsy and a whole variety of things, that if you know your human genome, you'll be able to say, this is what's going to happen to me if I get this drug or this treatment. Copy number variations is another form of genetic variation. And I've already talked about autism and schizophrenia. Increasingly, we are able to take a cell off an egg and find out whether that egg is abnormal. And although at the moment it destroys the sperm to look at the genetics of the sperm, we can do that. Detecting abnormalities in the embryo has been possible for more than 20 years. And now with new techniques, it's going to become even more common. And then the next big thing that's really going to flush through into practice is the ability to look at the baby's DNA in the mother's blood. And this has been known for 10 years. But tests will soon become available that within the first 4 weeks, say, of pregnancy, that we'll be able to take blood from a mother and find out the sex of the baby, whether it's got Down syndrome, and a whole variety of other things. There's enough DNA going around a mother's blood early in pregnancy to be able to work out the genetic composition of that baby. Let me tell you something we're doing in Adelaide. So Professor Decker and Claire Roberts are part of a worldwide consortium called SCOPE. And this is 7,500 couples who have agreed to give their DNA very early in pregnancy, and then to be followed through pregnancy with a vast number of blood tests, ultrasounds, etc. And through using this huge biobank, we're going to be able to predict before people get pregnant and very early in pregnancy what complications are going to occur in pregnancy and later. So we're going to discover new molecules. We're going to be able to come up with key predictors for future health and success. And you'll notice that bioinformatics, that I'm going to talk about in a minute, is critical to this. So here's what's likely to happen, that you get couples who have preconception assays. You'll be able to say to them, if you get pregnant, you've got a 50% chance of getting preeclampsia or diabetes or of this particular congenital abnormality. And so people are tested beforehand. They're then tested early in pregnancy. And through a whole variety of proprietary algorithms, you're going to be able to work out whether this is a high-risk patient that needs to see an obstetrician, or whether it's a low-risk patient who perhaps can deliver under midwifery care or some other low-risk option. So for fertility treatment, this is coming. We're going to be able to work out people's DNA well in advance of them trying to become pregnant and advise them what things they should be doing. Another example, this is Alistair McLennan, and a young boy, Matthew, who's got cerebral palsy. And for many years, we've thought that cerebral palsy was because the obstetrician was neglectful or the midwife didn't do something right. And vast quantities of money has been paid out for medical negligence. We now believe the majority of cerebral palsy is not due to events that occur late in pregnancy, but probably occur early in pregnancy. And Alistair's got the largest number of cerebral palsy patients in the world in a register, and is now looking at genetic contributors, and has got a number of very interesting things that are coming up that say to somebody, you've got an increased risk, not an absolute risk, of getting a baby with cerebral palsy. And these are the sort of things that he's doing. And then there's testing embryos. And back in the 1990s, we were the first ones to take a cell off an embryo in Australia and sex it because it was associated with a disease that went with boys, and to get a live birth, and also cystic fibrosis. And now in Melbourne and Sydney, you've got an amazing concentration of skill to do this. It used to be that we just did a PCR, which was a single test for one gene. Then we went to trying to colour the chromosomes, and that proved to be not very valuable. And there's now a technique in which Leander Wilton here in Victoria is one of the world leaders, called Comparative Genomic Hybridization, where, and I won't go into the details, you can actually compare one cell from an embryo with what it should be. And this is an example that here are all the chromosomes of an embryo, and the result should be between these red lines. And you'll see here that there's a result for chromosome 18 that's outside of that line. This is an embryo that's got an extra 18 chromosome. It's also got an extra X chromosome compared to a male, therefore it's a female. Now these techniques are going to allow us to look at chromosomes and abnormalities extremely avidly, and it's going to be widely used. Whether it will be wisely used is one thing, but it's going to be widely used. And then finally, let me just talk about where things can go wrong. This is a diagram of showing the eggs developing, the sperm developing, and then the embryo developing. And there are two critical areas here where something goes on in the DNA. So the sperm and the egg DNA don't get changed, but they actually get attached to them things that lock onto a particular gene. And these locks stop that gene working. And this is what we term epigenetics. So the environment can put a lock on a gene so it doesn't work. It's still there, and if the lock's removed, then it can work. But basically, our cells are locked from the time that we are an advanced embryo, and they don't get unlocked again, but with two exceptions. When the sperm and the egg are formed, you get this unlocking. And if there's an adverse stimulus at this stage, that will affect that genetic component for the rest of that baby's life. And it also happens with an early embryo. And therefore, if we're really concerned about looking after the quality of our DNA and fulfilling our genetic potential, we've really got to be concerned about how we're producing eggs and sperm and how we're producing embryos. And there is an ongoing concern in IVF, particularly in this area. And again, Melbourne is a world leader. Let me just illustrate that. Let's say that this is what a cell should look like, and these are the normal locks. And to get health all the way through your life, you keep those locks in place. But if something goes wrong early in life as an embryo or as a sperm or egg, you take off those locks, and over the course of your life, disease develops. At the very earliest stages of life, if we don't understand this process, many of the things that we're doing in reproductive medicine or people getting spontaneously pregnant under various influences in the environment may have very, very long-term effects. And this is the science of epigenetics. And these are twins. And twins, although they've got absolutely the same DNA if they're identical, they actually have different expression of those genes. I have a wife who's a monozygous twin. She has multiple sclerosis. Her absolutely genetically identical sister does not have any multiple sclerosis. And this is true for cancer and many other things. They've grown up in the same environment. They have the same DNA, but something has changed along the way. We have lots and lots of animal data that this operates as well. These are animals called agouti mice. And you'll see that they've got different coat colors. They've got identical DNA, but if you expose the mother to folic acid, which is a common vitamin, you can change the coat color. Chris O'Neill in Sydney, who's a great embryologist, has shown that if you change the culture conditions of these embryos in IVF culture, you can change the coat color. So by changing IVF culture conditions, you can change an epigenetically affected gene. So what are we doing in our reproductive medicine labs? We don't know. The data in humans is not very worrying at the moment, but only time's going to tell. Then the information revolution. We have huge amounts of data now handled by old computers, new smartphones, Facebook, Twitter, Google, and our patients are incredibly able to access all this technology, often ahead of any doctor. We use information technology at the earliest stages of life by looking at developmental origins. So David Barker used information technology to link heart disease with birth weight. Michael Davies, who works in my area, and Jane Halliday in Melbourne, is able to link perinatal IVF databases, cancer, anomalies, pregnancy. In South Australia, I don't know if you can do it in Victoria, you can get the police records, you can get education. So you could theoretically see what culture you did on an embryo and what impact that had on education or criminality later in life. Huge potential for abuse and non-abuse. We're doing very large cohort studies of people with reproductive disorders. Bioinformatics is absolutely critical to many of the modern assessment techniques like I've shown you before. We're developing new sensing technologies as well. Tanya Munro, who's an incredibly talented young physicist, has now designed fibers in which it has more than 160 channels in them. And through each channel, she can answer a question. What is the pH? What is the oxygen consumption? What is the level of a protein somewhere? We can actually put these inside an embryo and measure things inside an embryo. So these are brand new sensing technologies. These are slightly older sensing technologies looking at the culture media. And I won't go into detail on that. And through collaboration with Lois Salamonson and others here in Melbourne, we're looking at what's going on, how proteins and genes are expressed in the uterus, embryo assessment, looking at new molecules that may improve implantation. So information technology and many of the genomic revolutions are coming together. And finally, David Gardner and others in Victoria are really good at automating IVF, doing IVF on a chip, so that embryologists probably will become less needed than they are now, and they're highly needed at the moment. So we can get a lab on a chip, we can do very rapid 3D ultrasound, lots of new technologies. Okay, so how does this impact on lifestyle and fertility? There was a paper by Hassan and Killick, where in United Kingdom, they went to an antenatal clinic, people who'd got pregnant, said, how long did it take you to get pregnant? And then they took a history from them. And these were the things that they considered negative. And as women had more and more so-called negative lifestyle factors, it took longer and longer for them to become pregnant. And some of these are quite strange, but that's what the paper said. We also now know that smoking, different forms of infertility treatment, etiology, high body mass index, poor socioeconomic status, all have a bad impact. Interestingly, Asians have a lower IVF pregnancy rate than Caucasians, despite being socioeconomically exactly the same. And above all, we have age. So age is the fundamental, most important predictor of success in natural or IVF pregnancies. And I've calculated here from Liz Sullivan's data on IVF, that for 1,000 cycles that start, here's the chance of that woman having a baby that's going to go on. And you'll see how dramatically it drops from, if you're under 30, at 342 per 1,000, down to 109 per 1,000 for 40 to 44. So all of these factors are potentially preventable, apart from obviously ethnic factors, but they're things that people need to know about and often don't know about. So let's look at weight. This is a chart that shows body mass index, which is a measure of people's fatness. And a normal weight would be between 20 and about 26. And you'll see here from 120,000 nurses in the United States that as the weight went up, so the chance of lifetime infertility reduced. So once you get above 26 BMI, your chance of infertility goes up. This is the chance of a woman having abnormal periods. It also goes up, but not to the same extent. So it's not due to not having periods. There's some other factor. I thought you might be interested just to see what eggs look like when you feed them. I'm not a great shopper, but I do notice you can buy omega-3 enriched chicken eggs and things like that. Well, human eggs and mouse eggs, and these are mouse eggs, can be fattened. So this is from a mouse that was given a week's high-fat diet, and this is a stain for fat, and I think you'll see the difference. And we're looking at human eggs and seeing exactly the same thing. And then mitochondria are things that are found in the egg that provide energy to the egg, and this is what the mitochondrial strain should look like. And if you put them on a high-fat diet, that's what it looks like. So we can change eggs at will by changing diets. What about men? Well, there are lots of men who are overweight, and Michelle Lane and Hassan Bakos, who are in our institute, looked at male BMI and showed decreased pregnancy rates and worse embryos. So this is their data, and I'm sorry, it's a little small to see, but this is normal to overweight men going through IVF. And you'll see here that the live birth rate was 41% if you were a normal weight man, and 12% if you were an obese man. And you can also see this with embryos. So this is normal, overweight, obese, and morbidly obese. And this is the chance of having expanded blastocysts, which are the best embryos. And you'll see that it's half if the man is morbidly obese. Now, this is a fantastic piece of work that's been done by Michelle Lane, Julie Owens, and others in our laboratory. And I just wanted to show it. They took mice who were normal weight and gave them a high-fat diet and made them fat. They were mated with normal weight mice. And the females that were born were obese, had increased insulin resistance and increased tendency to diabetes. The males were not increased, but also had those metabolic parameters. They then put these animals on normal diet and mated them with a normal mouse, normal weight, and you'll see that the male mouse has still got a problem and the female still has a problem. Now this is just coming through overfeeding a man. It's not changing the DNA structure, but it may be changing the function. And then if you look at the male mated with a normal female, you get a very large female with insulin resistance, and you get a large male as well. So if this is true for humans, a fat male is a very dangerous person and should be avoided. There are lots of other things in our environment. There's lifestyle issues that I've put here. There are potential environmental issues. And I need to move on a bit. So what are we going to do about this? Well, I would like to suggest that this is a way that we should go. We need to set up individual advice through doctors, midwives, and nurses for patients. And we need to take collective action. And that might include folate, added to bread, vaccinations, iodine, etc. So for individuals, we need to get a proper history and investigation that looks at genetic and other diseases so that people can plan before they get pregnant. For communities, we need to make sure that we've got occupational health and safety procedures and that we're doing the right thing by giving advice to people. And ultimately, we need to have proper preconception programs, which to the best of my knowledge do not exist in Australia and probably nowhere else. About 20 years ago, we started a program called Fertility Fitness, where we took women who were not getting pregnant naturally or on IVF, and through a 6-month program, we showed that they could get pregnant very rapidly. And John McBain and others have introduced this here in Melbourne. And then there's another program I'll show you about. So this is a randomized trial that we did as part of our Fertility Fitness program. This group went through the program, this group didn't. And you'll see that after 18 months, that 3 times more women were pregnant by going through this program. And that was both through IVF and spontaneously. Diet can also affect IVF outcome. So this group have recently published looking at what women eat when they go through an IVF program. And if they're on the Mediterranean diet, they have a much higher pregnancy outcome than a normal diet. And then this is a program that we're running at the moment, which we call the FAST program, where every couple has an individual interview with a nurse for an hour. And all of these things are systematically covered. A report is then generated, which goes back to the doctor and to the patient. And the patient is then given assistance to change. And so this is what we try and do, is give awareness to patients and doctors, do an individual interview, plan what we term a reproductive health pathway, give adequate advice, information, and provide resources. Let me give you an example of what we try and do. So when someone comes with infertility, they have a consultation for medical and lifestyle. We look at their infertility etiology. We look at the lifestyle factors that they've got, the lifestyle intervention, and clinical intervention. And the aim is to produce a healthy baby. So we try and tick the boxes on each one of these on patients coming through. And so I'll give you an example. Patient with polycystic ovary syndrome doesn't ovulate. So we go through lifestyle factors such as obesity, poor diet. We look at interventions, and then she gets pregnant spontaneously. We have another person who's got male factor infertility. They are also obese and need things sorted out. They go through a lifestyle program, but they do need advanced technology, and they have a baby. So I want to propose that we have reproductive life plans for everybody, and it sounds a bit prescriptive, but if we have financial plans for people, why should we not have a reproductive life plan? And that goes from the time that somebody first has a period or goes through puberty and covers things like contraception, vaccine for cervical cancer, pap smears, etc. And it's updated as circumstances change. People need information and they need to know where to get it, and I think it should have government support. And then we need another process, which is a preconception interview. And this is where, before people choose to have a baby, they have access to facilities and information, that they can get specific information they can act on, that they get expert advice, and again, that this is adequately funded. So how should we invest in periconception? Well, we've got a number of outstanding institutes that are doing great work. My institute, Prince Henry's Institute for Medical Research, Monash Institute for Medical Research, Murdoch Children's, among many others. There are a number of very outstanding groups that are not part of institutes. And we have excellent epidemiology and public health. And all of these we can put towards the earliest stage of life. And then we need to take what we already know and translate it into action. We've got a lot of knowledge, but we're not getting it across to people. And so we need some of these things. So just to tell you about the Robinson Institute, we have 4 major centres, and due to lack of time, I won't go through detail what they all do, but they cover from molecular biology through to public health and epidemiology. We have a very large staff. We have a lot of very senior, experienced people, and we integrate with all the IVF units and the public hospitals in the state. We've got great science, clinical medicine, public health and epidemiology, and translational medicine. And again, for lack of time, I won't go through details of that. And basically what we're trying to do is to provide a strong evidence base. We're trying to influence leaders in health and politics to put this evidence base in place. We want to inform patients as to what is available to them. And we want validated intervention programs that actually work. So let me conclude, and I'm sorry I've gone a little over time. I believe that healthy development in any society starts around the time of conception, and that a small investment at this stage is really worthwhile. We need very good technology, but we also need good health policy and Medicare funding. We do need to involve society. This is not for doctors and nurses and health professionals. It's to get the whole of society involved. And we need personal commitment from people who are planning a baby. I think we need preconception programs across the state, and it's not in reproductive medicine units. It's available everywhere. We need to be communicating with people who may plan a pregnancy. I believe, and this is controversial, that every assisted reproductive technology unit should have a mandatory lifestyle program as part of their accreditation, and that will bring questions. And then research focus should be focused on healthy development. So in conclusion, investment in early life makes sense in every way for economic, health, social, and human capital. And Amartya Sen was a Nobel Prize-winning economist who recognized that investing in developing people was freedom. And I believe for the sort of people that we deal with who want to have children, that this is a fantastic freedom that we give them by giving them information. And I'd like to thank those who've helped me on my journey. Thank you.